Hi, everyone, and welcome to the afternoon scientific track for the National MPS Society's 34th Annual Family Conference virtual. It is a distinct honor to welcome the next presenter, not only because of who he is and what he's done for many years for our kids with MPS, but also because he is our Chief Scientific Officer at the National MPS Society and joined us just a few short weeks ago and relocated to Durham, North Carolina. Let me tell you a little bit about Dr. Ellinwood. He was Associate Professor in Comparative Medical Genetics Research at Iowa State University since 2004. He trained at Colorado State University in physiology, after which he pursued residency and postdoctoral training in veterinary medical genetics and gene therapy at the University of Pennsylvania during his tenure from 1998 to 2003. And the Gene Therapy Laboratory in Nantes, France from 2003 to 2004. Again, as I mentioned, he had been at Iowa State University, and it's just a coup for us to have him join the team. And he's going to talk this afternoon about gene therapy and pharmacological considerations. We welcome Dr. Matthew Ellinwood.
Thank you so much, Terry, for that introduction. So today I will be talking about gene therapy in an overview sense. Last year I concentrated mostly on traditional gene therapy, and this year I would like to focus on a sort of new and up-and-coming technology called gene editing. And so that will be the most of the focus, and a lot of this is sort of new technology and may not have application immediately, but certainly we hope it will in the future. So to give you some context, we'll be talking about genetic disease, gene therapy, and the limitations of these approaches and the types of these approaches. I'll discuss the difference between gene therapy and gene editing. They are not the same, although there are some overlaps. We'll also go into detail on gene editing techniques. What is the state of the art? What are current applications? And what is on the horizon? And I'm hoping we'll finish early enough that I'll be able to take some questions. If you write your questions, definitely I'll be able to address those later on, if not today. So genetic disease and gene therapy. Genetic disease results from some kind of sequence change with disease-causing downstream effects. So in the MPS and ML disorders, we are only seeing DNA changes in what we call a single locus for a gene. So these are not complex genetic diseases. There's only one gene locus involved per disease. Most are autosomal recessive, of course, except for MPS II. Most, but not all mutations affect protein production at what we call the post-transcriptional step, right? So I hate to take you back to high school biology if that causes some trauma, but when we go from what we call the central dogma of biology, we have genetic transcription, and that leads to genetic translation into some kind of protein. In biology, that's basically going from DNA to RNA to protein. Most of our disease-causing mutations will affect not necessarily getting an RNA molecule, but something affecting the translation process. So what does this look like when we're actually talking about a cell? Here's a cartoon of a cell. We've got a chromosome and a nucleus. We get transcription, and we use that term simply because you're writing down a different code in a similar molecule of nucleic acid, an RNA molecule that's single-stranded. Pretty much the same sequence with some minor changes. One of the base pairs is a little different, right? So we've got that, and then that will get exported outside the cell nucleus, and it will be translated by these little machines, basically, that will read the mRNA sequence and create a protein. In the case of our diseases that are either secreted or membrane-bound proteins, this occurs while these ribosomes are connected to a membranous sort of sac within a cell called a ribosome. We get these nascent proteins delivered inside the ribosome where they're matured. So again, most of our mutations are going to affect RNA in some way. Maybe the stability of the RNA is affected. Or there may be a problem in the translation of the RNA, turning it into the new language of protein from nucleic acid. And some of the mutations could, of course, involve the protein function itself. So, treating genetic diseases. You have a gene therapy, it's a genetic disease, you correct the gene or you deliver a new copy of the gene, you've got it treated, right? Well, yes and no. There are some considerations to take into account. Is the gene for a soluble version of a protein, or is it for a membrane-bound protein? In other words, are the cells that we treat going to be able to produce a protein, release it into the plasma where it can be taken up by other cells? Or when we gene therapy tissues, is it a membrane protein that's only going to stay in the treated cells and not go anywhere else? So that will influence which of our diseases we can theoretically treat with gene therapy. Also, what degree of therapy can we get? What % of normal enzyme? What % of normal enzyme in which cells? Whether we're treating connective tissue cells or liver cells or brain tissue cells, that can have a big difference. And of course, from what age? How soon are we able to get cells treated in a patient? Can all have a big effect. So as part of my talk, I would also like to introduce some landmarks in the studies of our diseases, sort of like lighthouses that really cast, I hope, a long beam both into the future, what we should be looking at. And one of those is a study on neonatal treatment of MPS 7 dogs using a retroviral therapy. So this therapy, which was pioneered by Dr. Mark Haskins at University of Pennsylvania, led by Kathy Ponder at WashU in St. Louis, came out nearly 20 years ago. And it was treating MPS 7 dogs basically from birth. These animals were injected one time with a retroviral gene therapy vector that was able to give them very high levels from the very earliest days. So this is day 50. In fact, within the first week, we had very high levels of enzyme. This is, you can see 100% of normal is the green dotted line. And we have some animals who were nearly 100 times normal. This is a log scale. So that 100% of normal, 1,000% of normal, 10,000% of normal, 100 times normal levels in at least one of our dogs. And this was an animal treated within the first week of life. They had these levels of enzymes. So this is sort of, in my mind, in a large animal model, the best example of what we can get with an enzyme being produced in the body from some cell in a liver in this case. So how well were we able to do? Well, the animals, for a disease where you are having to euthanize animals at 9 months, there were animals that lived out to 12 years. And this animal that I'm showing you now, Preston, was one of those. So it was a substantial treatment. But even if we were getting enzyme levels 100 times normal within the first week of life, you can see that there were still serious orthopedic problems. So there was a good paper written on follow-up on the orthopedics of this disease, and a conclusion was neonatal gene therapy with an retrovirus to MPS 7 dogs can reduce many qualitative abnormalities of bone and joint disease. Nevertheless, skeletal disease was not completely corrected, and disease in the cervical spine remained severe. So we still, even in the context of really successful gene therapy, have a lot to do to understand the disease in certain tissues like bone and to figure out how to address that treatment better. So the limitations aside, what else is a potential benefit of gene therapy? There's still a lot of proven and future potential. And when we think about gene therapy, you can understand bone marrow transplantation, which is one of the oldest and now standard of care therapies for MPS 1 in the form of hematopoietic stem cell transplantation, as really a gene therapy. You are delivering 1 or maybe 2 copies of a gene inside transplant cells. It's also a cell therapy, so it's also in some respects our first combined treatment, which is getting to be a mode of treatment in experimental animals that we're thinking is going to be necessary for a lot of these diseases, combining different approaches. So by gene therapy, I mean the addition of genetic material to cells or tissues that treat some disease. These can be genetic diseases, not always. Gene therapy is being used for cancer, for example. In the case of MPS and ML disease, this usually remains replacing the faulty gene. We're going to add an additional copy in some tissue. Genetic material is usually delivered via genetically engineered viral particle. We call these viral vectors. I don't like to call them viruses. They're viral vectors, as it were. These particles can deliver genetic material to cells and tissue, and they come in two main flavors. There is something called an adeno-associated viral vector, referred to as an AAV vector. That is used both experimentally, and it's also being used in clinical trials now. You'll hear from Abiona, who uses an AAV vector. It's approved as a therapy for some genetic eye diseases. It's a real workhorse out there. The other approach involves two types of retroviruses, retro and lentiviral vectors. And again, these aren't viruses, they are vectors. And this has a transgene that gets actually integrated into the genome. So AAV vectors, the transgene doesn't integrate into the host DNA. Lentiviral and retroviral vectors, the transgene will integrate semi-randomly into the genome. And there are both advantages and drawbacks to this approach. So instead of gene therapy, which I covered last year, we're going to really talk about gene editing. So what is gene editing? I consider it a subclass of gene therapy. I think there are some gene editors out there who think it's its own thing. This involves a targeted editing or changing of the genome. And in contrast to the integration of retroviral or lentiviral vectors into the genome, that was pretty random, although there were areas where it liked, the kind of areas where it liked to go. These additions or editing of the genome, these are precisely targeted. We're zeroing in on definite sequences and changing them. So who are the players? What are the kinds of approaches that we may have heard about that are involved in gene editing? And these are just different technologies in molecular biology. And there are three main classes now that you may have heard about in this area. One is a zinc finger nuclease. Nuclease just means it cuts DNA. Zinc finger is a description of the structure of the protein that identifies the DNA polymorphisms. We have something called CRISPR-Cas9, and we'll talk about what that means. And we also have something called base editing, which is the new kid on the block, so to speak. So let's back up again and think about what diseases are theoretical candidates for gene editing therapy. Given the current constraints of this approach to therapy, which is really the big constraint is efficiency. If we use these, we are looking at changing fractions of percentages or single percentages of cells within a given tissue. And we are not, if we think about correcting a mutation, if we can get the liver to produce, 1 to 4% of enzyme, that's just the liver. It's probably not going to treat the rest of the body if we're talking about correcting the native gene. So we could get some cross correction, but actually going in and correcting these individual genes is not going to be very productive. But theoretically, all of these diseases, because they have soluble proteins, are targets, if not for direct gene correction, for some kind of gene therapy. Diseases that involve membrane proteins like MPS3C and ML2 and ML3, they are going to be less amenable because those gene products, those proteins aren't freely soluble under most situations, and we can't create a production factory within a body as in the liver. What about mutation types? As long as the mutation is fairly small, we can think about correcting it for the most part. Some mutations, large deletions where the whole locus, very large portions of DNA, we're not going to be able to correct those by editing because there's nothing to correct in those particular situations. So let's talk about zinc finger nucleases. How do they work? So you've got a protein, a zinc finger, and it cleaves the DNA, and then you have an addition donor DNA added, and it uses something called homologous recombination and the cell's DNA repair machinery to integrate that corrected part of the gene in the blue bar into your targeted gene replacement area. To think about this a little more clearly, the zinc finger is the portion that is in blue circle, recognizing the DNA. The nuclease, which is the cutting portion, is there. You have two different ones to recognize sequences very specifically. They will cut an area of the DNA. You have your transgene in there, and cell machinery that is active in that cell will hopefully repair it using the supplied corrected gene homology template. So that's the theory of gene correction, so to speak, through gene editing. So the current limitation on zinc fingers, they're not efficient. And the cell type that we can use may be limited by the degree of homologous recombination that's present. So how do we overcome these limitations? Well, if you think about combining a bunch of clever strategies, you can do that. The liver is a great protein-producing organ. Target the liver. Decide not to repair the disease-causing DNA change. As I said, that may only give you 4% of normal levels in the liver, and that may not be anywhere near therapeutic enough to treat the whole body. But you can use these zinc fingers to introduce a normal copy of the gene, in this case, a DNA version of that messenger RNA called a cDNA. You introduce that version into another gene in the liver, in this case, an albumin gene whose promoter will drive that gene's expression. So the promoter is the portion of the gene upstream of the protein coding area that will tell the gene when to start and how much to produce. And in the liver, the albumin promoter is a workhorse. It is pounding out albumin all the time. And if we can introduce our cDNA downstream of that promoter, our cDNA, our transgene, will benefit from all that extra promoter activity. And indeed, this very clever approach is the basis of what Sangamo Therapeutics is doing in their Empowers study and in their Champion study for MPS 1 and MPS 2. Now, we still have to get these zinc fingers and the corrected the cDNA with homology arms into those cells. So we're doing that with an AAV vector. In this case, it's something called an AAV26. It means the exterior portion of the virus comes from a form of AAV called 6, number 6. And the interior DNA portion that's included with our transgene comes from an ancestral form called 2. So most all of our vectors are going to have those little bits from AAV2. And then the second number will tell you which serovar it is, what variant of capsid protein. So these are designed to deliver these portions of the cDNA and the zinc fingers directly to the liver and lead to the introduction of the MPS 1 cDNA for iduronidase or iduronate sulfatase for MPS 2, and to get high levels of production from the albumin locus. The animal studies for this were incredibly promising. Preliminary results have been presented last year, indicate possible dose effects in the enzyme levels, dose effects associated with the level of gene therapy introduced. There were also some issues, at least one case of immune targeting of the liver. The company is working on second generations of zinc fingers to improve both targeting and efficiency. Theoretically, there is still some hope for this approach in terms of they did detect enzyme, but we need to think about maybe more efficiency or a higher dose in this case. So we'll look forward to seeing what that next chapter of therapeutics looks like. So that's zinc finger technology. What about CRISPR-Cas9? Many people may have heard of this. It's in the news. What is this all about? So to review the natural history, etc., CRISPR is an acronym that stands for Clustered Regulatory Interspaced Short Palindromic Repeats. Basically, these are bits of foreign DNA that have been chopped up, repeated, and introduced into bacterial genomes. So the whole CRISPR-Cas9 system, it's like a primitive bacterial immune system that chops up viral DNA from an invading virus. And we've been able to sort of exploit this and further genetically engineer it so that we could go in and instead of identifying viral DNA to chop up, we can program this to identify particular DNA sequences we want to cut. So it's composed of a protein, a nuclease called Cas9, which stands for CRISPR-associated nuclease. And in this case, Cas9 is the best one known or used, but there are now more than 20 of these with different numbers. They can be found in different bacteria. They're discovering all sorts of potential diversity in this area that will help diversify the utility of this whole class of gene editing tools. Overall, it may be less programmable than zinc fingers, but it is much more flexible in other ways. There are always concerns both with zinc fingers as well as with CRISPR-Cas9 systems or other similar CRISPR systems of what we call off-target cutting. So these are cuts that may be introducing mutations in places where we did not want them to be. There are tremendous improvements being made in this area, and it will be something to follow so that when it comes to clinical practice, we have a high degree of comfort that they are going to be safe. It is a very active area of research, and I tell you, this is one of the things that's most exciting to follow because there's so much happening. If we think about how this looks in terms of a molecular model, here you've got your double-stranded area of DNA. This little thing is a piece of RNA we call a guide RNA, the yellow piece there. And it is programmable. We can put in our mutation sequence that we want to correct. It will bind to that sequence. It will help open up the DNA with the help of CRISPR, and CRISPR will cut at some particular area here. So the red blob is the CRISPR protein. The yellow is what we call guide RNA. It's not really part of a CRISPR sequence, but that's kind of the function that it's serving in this context when we use these as engineering tools. So it's now an application that targets mutations or otherwise edits genomes. Many small mutations are potentially targetable, but again, large areas of mutations may be very difficult for us to correct. It does rely on the body's ability to repair a DNA break the same way that zinc fingers do. We have to provide some kind of homology that has a corrected DNA sequence that the cell will use as a template to help repair itself. Now, these mechanisms of homologous repair, there's machinery associated with them, and they may not be in all cell types equally. So that may be another restriction. So how's the best way to target cells for CRISPR editing, and where? Or do we want to do it in a petri dish? Do we want to do it in vivo? In fact, at this point, it's not efficient enough for our diseases to think about doing it in vivo. But there has been a genetic disease, sickle cell trait, very different from our diseases. It's a disease of red blood cells. It is similar, however, in that just small percentages of improvement could help patients. So in this case, they took bone marrow cells from patients, and in ex vivo, in a petri dish, they repaired the patient's stem cells using CRISPR technology. And what they did was they turned on a second hemoglobin gene by mutating a repressive element in the promoter. They turned on the fetal hemoglobin gene. And the results from that have been extremely promising. And it's been done in sickle cell trait and also for another red blood cell disease. So this kind of approach has some promise. So applications of in MPS and ML, the small increases in enzyme that will come from repairing the patient's mutations in the liver, for example, are probably, it's going to be too inefficient. And as I said, treating anemias is different. We can only have to treat a couple percentage points to help them. It is, if we think about combining it in an approach similar to Sangamo, replacing CRISPR for zinc fingers, that approach could be very helpful. Also, if we think about using it to transform hematopoietic stem cells ex vivo, where we introduce a very highly expressing gene, that would be another approach. Or if we're trying to treat retinal diseases where the tissue is very small, those sorts of approaches may be on the horizon. But again, it'll be on the cell type, also involving the machinery of repair, in addition to which cell we're thinking about. So we come up to another approach now, and this is even more interesting, and this is very, very early days. I don't know that we're talking about clinics anytime soon for this sort of approach for our diseases, but it's important to be thinking about. It's a very hot area of research. So in CRISPR, what you're doing is cutting the genome, and you're relying on homologous templates of normal sequence to be used in the repair. In base editing, we don't cut the DNA. We go in and we discretely change one aspect of one single DNA base pair, and that can have the effect of changing a CG base pair in the DNA sequence to a TE or TA base pair. That's so-called cytosine base editors, CBEs. We can also change another type. We could take an AT base pair and turn it into a GC base pair. The sites are targeted using a CRISPR system, but it has a defective Cas nuclease that can't cut. And instead of having the Cas nuclease cut, we have a cysteine base editor attached to the Cas, and these are enzymes, and they will catalyze a base change. So you will see it doesn't change all potential base pair mutations or base mutations, but it can target about 30% of known human pathogenic mutations theoretically. So how does this look if we look at it sort of in cartoon form? You've got a sequence and it's CG in a mutant gene sequence. And you develop a guide RNA here with your CRISPR that's in gray, and you've got your cytidine deaminase, which is the name of the enzyme that will change that C. It changes it actually to a uracil, which is a different kind of base. And then when this DNA goes into replication, because that's going to be mismatched with a G, it will actually turn that into an A, and then that gets turned back into a T. So you get a base pair change from CG to TA. Again, you haven't broken the DNA, you've just gone in and made one discrete change that the body turns into a TA pair. Incredibly clever approach. We also have something called, base editing technology for the MPS and ML. This is a promising avenue, but it's still currently under development. There are going to be issues of safety, how efficient it is, because we are going to need it to be widespread throughout the body to create corrections in meaningful levels of tissues. And we're going to make sure the specificity is good, so we're not getting off target cutting. And then there are also problems with delivery. How are we going to figure that out? But again, these base editing techniques only target a certain group of mutations. So how are we going to make sure that we get all of them? Well, there is a new next generation approach called prime editing. And I'm not going to get into any detail on this, but prime editing potentially can target all possible base pair mutations or base mutations in the body. It can correct all 12 of any potential mutations that are single base changes. Again, it needs to be a single base polymorphism. So overarching concerns for all of these approaches include efficiency. Right now, in vivo, we are at fractions of percent or single percentages of base or of mutation transformation. And translating that to in vivo gets tougher. There is, as I said, huge potential for these approaches to employ things like transforming the liver at the albumin locus, or there are also what we call safe harbors within the genome, areas that are very easy to insert DNA sequences into. And if we could insert a highly expressing so-called DNA cassette, a good promoter in front of a cDNA in one of these safe harbors using this gene editing technology, it would be basically a variation of the Sangamo approach. Those sorts of things are important and potentially useful. The idea of also of inappropriate cutting, or what we call off-target cutting or changes, that's a concern. It's a concern for a number of reasons. We may produce neoantigens, new antigens that the body has not seen. If you're correcting a gene that the body's never seen, that itself is going to be a neoantigen. So a lot of the old concerns about immune response also persist for these approaches to therapy. So in many respects, our concerns are very similar to what they are for all of our other approaches to delivering exogenous protein. So wrapping up, how do we think about gene editing? The promise of gene editing as a basis of therapy still confronts many of the old issues. What is our window of opportunity? How soon do we have to get treatment on board? Hopefully with the developments in newborn screening of the sort that Dr. Burton presented, we're going to have the ability to get in and get treatments to patients at that very earliest possible stage. So hopefully we'll see great strides in that area. Again, what level of therapy do we need to prevent diseases? How successful do these need to be? And animal model work will help answer some of that, but a lot of it's going to be trial and error in some respects. And then, of course, how are we going to get enzyme to hard-to-treat tissues in the central nervous system and in the skeletal system? So even though we've got a new tool, we've still got all of these old concerns to be thinking about. And again, I think the biggest innate constraint to gene editing technology is the efficiency at this point. Maybe soon we'll be getting much more efficient, but at this point, we're going to be limited to treating very discrete therapeutic targets where there's one small area of tissue, say, for example, in a retina, or we're talking about altering a locus a particular area of the genome, like the albumin promoter in the liver or a safe harbor, that we can use to create sort of a factory to produce more enzyme. So that may not be very hopeful in some respects. I talked just a lot about constraints and concerns. However, these tools for the researcher are revolutionary. So the speed with which we are now able to make progress on all sorts of questions using things like CRISPR, Cas9, this application of it has been incredibly valuable. We are making progress much, much faster. We can create a transgenic animal model within months when it used to be years using this kind of technology. So there may not be benefits that you guys will see in clinic, but from the researcher's standpoint, their ability to make really quick progress and help bring things deliverable to clinic are really improved by this kind of gene editing technology. So with that, I ended a little bit early by design so I can continue to take some questions. And let's see here. I need to figure out how to get to questions and see what I can
ask you the questions if you'd like, Dr. Ellinwood.
So that would be great, Terry.
One of the questions that we have is, do you believe treatment efficiency to be impacted by uncertain durability of expression in pediatric populations?
So there are a couple of things that will dictate how persistent a gene therapy is. One of those is, will the cells persist that you have transformed? Are they going to persist for the lifetime of the patient? Will the DNA that you've introduced persist for the lifetime of the patient? I can tell you what we know about some of these diseases and approaches to therapy. If you're going to be transforming the genome, so this is a therapy that involves transforming hematopoietic stem cells, as is being studied at Manchester, to produce highly expressing enzymes via a lentiviral vector outside the body, I think those are going to persist, right? That sequence is integrated into the host's DNA. Those hematopoietic stem cells are going to be put back into patients. They should persist. Bone marrow transplants persist in patients, even though they're pediatric. So I think in some cases, yes, where you've got stuff into the genome, certainly. That example of retroviral therapy in the dogs, we had persistent levels and expression out to 12 years of life, and we didn't really see anything change. And that was an animal treated at birth. For things involving adeno-associated viral vectors, those DNA don't integrate into cells, and there is concern about it being lost over time. We do know, at least from experiments in eyes, that those vectors can persist for many, many years and don't seem to be getting lost. I think if the AAV vector persists after the first potential for an immune response, I think even if it's a pediatric patient, there's a good chance that it will persist for a substantial period of that patient's life. I don't want to go beyond that. There is something that we saw early on called promoter shutdown, where it seemed that the body sort of quieted down the promoter of a transgene. There's been a lot of improvement in our technology, so that is less often a concern when we're actually introducing novel genetic material into the host's genome. I hope I answered that.
I think you did. We do have another question. Someone would like to know, it's Dr. White, he'd like to know, could you please discuss the risk of hepatotoxicity in AAV-mediated gene therapy studies? An example is X-linked myotubular myopathy. And then how might this be avoided in the future?
So I am not precisely familiar with that particular example of AAV therapy. There are two approaches. Is there an innate toxicity of the vector that causes a deranged immune response? And I think there are approaches to genetically engineering the capsids or the coverings. Remember, I showed you that example of a vector that Sangamo was using, which is called an AAV26. So there are examples. I know there's a company called Lacerta in Florida, out of University of Florida, that Barry Burns involved with, where they're actually mutating the capsid of AAV6 or AAV8. It's a triple mutant capsid where they're trying to reduce the ability of these viral particles to cause an innate immune response that can lead to some of these very serious immune responses. The other issue is there an immune response to a novel antigen, a new gene being introduced? And that may be a gene the body ought to ordinarily produce, but the body's never produced in a patient who has a genetic disease. Those are still novel antigens, so to speak. That particular problem, I think, should be able to be managed by immune suppression that is designed to taper off over time. So I think this was a report that came off last year that I think you're referring to. I think there are approaches to tweaking with the dose and also the composition of the vector, identifying which of those amino acid residues on capsids may be triggering this response. Thank you.
The last question that we have, well, a couple more questions real quickly. One wants to know, George, would like to know if these technologies are going to be actual therapies or treatments for Hunter syndrome.
There is no reason that these approaches could not be used for Hunter syndrome as long as the allele does not have a giant deletion rearrangement, which is the case in some Hunter patients. Dr. Munzer and Burton would know better what percentage of those Hunter patients have large deletions. But as long as you have in terms of gene correction, as long as you have a smaller mutation that is amenable to correction, potentially yes. The Sangamo approach, where we're actually introducing a therapy to be overexpressed from a promoter in either a special site, a privileged site, a safe harbor we call it, or the albumin promoter, not only could that treat Hunter, there's a clinical trial ongoing to evaluate just that in MPS II patients. So yes, this approach absolutely could address the concern of the Hunter patient population.
Thank you, Dr. Ellinwood. That ends our queue for the Q&A, and you can continue on with your next presentation.
Okay, I'll have to figure out how to get to my next level of slides, and that's that one, and I think it's here. Somebody queued it up for me. So this talk will be also, in some respects, a lot of theory of how we treat diseases. And when I give you these talks, I'm trying not to just give you a lot of details and facts, but I'm trying to help you think about classes of diseases, types of mutations, and approaches that we can theoretically treat. And that's very much the case when we talk about drugs. By a drug, I simply mean, a pill or a medication that's infused. ERT is that's a drug therapy. We're talking about something that's not necessarily an enzyme replacement, so to speak, so other kinds of drug therapies. So I'd like to talk to you about the context of drug therapies, types, benefits. Many of these are all theoretical at this point, and they have discrete niches. And the thing that is making it difficult for us to develop them is our model system to screen certain kinds of drugs. I think that's the big hurdle, so to speak. If you think of it in enzymatic terms, we have too much energy that we have to put into a system because we don't have model systems that will make it easier. And I'm not going to talk about drugs for symptomatic therapy. That would be really the purview of the clinicians. I'm going to be talking about drugs that will in some way treat or address the root cause of disease. There are a lot of promising avenues, but there are also some limitations. This tends to bring up lots of questions, so hopefully I will finish in time so that you have an opportunity to get me some of your questions. And you guys can always feel free to email me at the Society with any questions about these sorts of things. So, whoops, did I lose? So, when I think about drug therapies that aren't symptomatic or aren't ERT, I'm thinking of drug therapies in two big classes or ways that they may influence a disease. One is going to influence translation of mRNA or some aspect of enzyme stability or traffic, which I just lump together calling enzyme function. Within that category, I've got sort of three sorts of classes that I talk about. One is pharmacoporone, which is a term, not I use it, some people use something else. It is basically a pharmacological form of therapy, a drug therapy, and it acts as a chaperone. Now, I'm not talking about an old lady to escort your daughter at prom night. These kinds of chaperones are molecules in cells that will help bind a protein to keep it protected. And in this case, I'm talking about it keeps it protected as it's being produced. A chaperone can also be used to stabilize an enzyme once it is produced. I call that enzyme stabilization. Also influencing this particular step in biology, the translation one, will be something called stop codon read-through drugs. Dr. Munzer mentioned that, and this is a potentially very attractive approach because there are two stop codon alleles for MPS I that represent a very significant percentage of the alleles in the population. So this could impact a lot of MPS I patients. So that's a drug that targets translation or some aspect of enzyme trafficking, stability, function. There are also drugs that may influence disease pathophysiology in some fundamental way. We're going beyond just treating the patient's symptoms. So you probably have heard about substrate deprivation therapy. That idea is that if your body has no substrates to accumulate, you won't have disease. In this case, it would be a substrate deprivation targeting glycosaminoglycans because that's the substrate that gets stored in MPS diseases. We also have drugs that may interrupt certain aspects of pathological cascades. So think back to that picture I showed you of Preston, the dog at 7 years of age. He had those spinal cord lesions in his neck. In that first week of life, as an MPS VII affected dog, before he had those sky-high levels of enzyme, he probably had some pathological cascade in his bone tissue that started that enzyme couldn't interrupt in. So figuring out what those cascades within cells are and interrupting them could be very helpful. There's an approach to manipulating vesicular traffic. So vesicles, lysosomes are just sort of bags of membrane within cells. We call those vesicles. And can we influence the cell's system so that it produces more lysosomes? Or, hey, maybe it even encourages lysosomes to fuse with the outside of the cell. That's another active area of inquiry. And then, though you won't hear any official discussions on this, I call this class the what the heck class. Scientists never want to say they don't know how something works, so they don't acknowledge this class. But there are some drugs for lysosomal storage diseases. We're not certain how they work, but they do. And the problem with that is these are hard to discover. And the one case where I know about it for Niemann-Pick C, it was rather a serendipitous discovery. But we'll talk about that, at least as a class, to think about. So drugs and how they interrupt or change translation or enzyme function. So most of the patients who are going to be amenable to these drugs, they're going to have some level of RNA, right? This will often, in many cases, be attenuated patients. And again, these are only potential candidates. We're not talking about real therapies that are known. This is sort of a theoretical approach. So the mutation must have a missense mutation. That means an amino acid has been substituted in the protein, or there have been an in-frame deletion. 1 or 2 or 3 amino acids have been removed, but the rest of the protein looks good. So that's a mutation that affects this portion. Some of these missense mutations may affect how well the protein actually gets translated. And that happens in a system called the rough endoplasmic reticulum. And we'll show you an illustration of that, how that happens. So these are 1 amino acid substitutions or small in-frame deletions that give you a potential protein, right? So that's one class that we can think about treating with pharmacoperones. Stop codon read-through, that's a class where the defect is at the RNA level. So if you have a stop codon in an RNA, like those 2 common mutations for MPS 1, that RNA is degraded in the cell for the most part. And so you don't really get to see it turn into protein. But there is a drug that can target that process. So let's go look at an illustration for pharmacoperone therapy. But first, let's talk about what kind of percentage of enzyme activity you may need to get some benefit. So I like this schematic just because it offers a good visual presentation of what kind of enzyme level we're talking about to get therapy. So if you look at this blue triangle standing on its end, the top is normal people with high enzymes levels. As you get further down, but above that line that says threshold, you may approach that area that Dr. Burton was talking about, where you had pseudo deficiencies. They looked low, but the people were still in what we call this normal range, right? But as the enzyme levels go longer, you start to creep into mild, attenuated, and severe forms. And that corresponds to how low your enzyme level gets to be, right? So for most of the cases we're thinking about for pharmacoperone therapy, patients are going to be in this probably mild to intermediate level as potential therapeutic targets. But if we're talking about stop codon mutations, they could be severe individuals. And depending on the drug's ability to normalize enzyme activity, even those individuals could see therapeutic benefit. So there were some questions early on in Joe Munzer's talk about how do we explain my child's phenotype? Some of it looks severe, some of it doesn't. We don't know. So one of the things I also, in my time here with you, is to give you a sense of some of the important literature that's been done out there. So there's a big concern and question about how do we correlate one patient's genotype to a phenotype? And this was an incredibly important study in MPS 1 done in John Hopwood's lab, headed up by scientist Susanna Bunga, who was visiting from Europe in his lab. And this is really a gold standard in my mind for telling us how much enzyme activity gives you a patient who has Shea versus Hurler-Shea versus Hurler. And as Dr. Munzer said, that's a spectrum, and it's an artificial spectrum, but at least to distinguish certain aspects of the phenotype, I think it's a useful construct. So this study was an incredibly difficult and rigorous in vitro characterization of the biochemical aspects of the disease in fibroblasts, and these came from patients where they knew the DNA mutation and they knew their phenotype. They were using ultra-sensitive, high-affinity radioactive substrates which are not the sort that are used in the diagnostic lab. So this is very much just a research application. They were also looking at radioactive sulfur incorporation in cells and also purifying enzyme with antibodies so that they could measure it specifically. And some of the take-home messages about what distinguishes a hurler from a hurler Shea from a Shea from a normal in enzyme capacity is telling. So another take-home is there's no way to do straightforward genotype-phenotype calculations unless you have a study, unless you have known mutations that are not novel. But the other take-home was when we look at catalytic capacity of normal iduronidase, and we use that to make percentage estimates of what gives you a hurler patient versus a hurler Shea versus a Shea. Hurler patients were 0 to 0.1% of normal enzyme catalytic capacity, and a Shea patient was as little as 0.4% to 7%. So for our diseases, our therapeutic target may be very low, but we still have a lot of challenges in using drugs, gene editing to get there. The other thing about these measurements is these values were present in patients from the beginning of life, so to speak. And all of our therapeutics, we are intervening, and we may be intervening at times when windows of therapy have not necessarily closed, but there's already been a cascade of pathology set up. So that gives you an idea of our target may be very low. A drug therapy, if it could induce normal enzyme protein levels that gave you 7 to 10% of normal enzyme activity, that could be a substantial benefit for a patient who was hurler or hurler Shea. So how does this look in the cell if we're talking about pharmacoporons? So this is a cartoon of a cell. The circle at the bottom, the yellow one, outlines those little squiggly forms inside the what we call rough endoplasmic reticulum, where mutant protein is hanging out too long, not folding, and it gets sent to a disposal factory called a proteasome. So if you have a mutation that leads to a protein getting put inside, getting translated into the rough endoplasmic reticulum, but the mutation doesn't allow it to fold properly, it will be degraded. A pharmacoporon is a small molecule drug. In this case, it is indicated by that little sort of circle that helps get that enzyme out of the rough endoplasmic reticulum. It stabilizes it. It allows it to go through and get matured and put into the lysosome. So scenario 1, a missense mutation and no drug, misfolded protein shuttled to the proteasome. Scenario 2, in the purple oval above, a missense mutant RNA and drug. The drug stabilizes the folding of the enzyme and it gets taken to the lysosome. So that's the theory. But can it work? Again, only some mutations are candidates. They must be missense mutations. And the affected mutation, it cannot affect the active site of the enzyme, where the enzyme actually does its work, right? Not all missense mutations are going to be sensitive to the same drug. Many of these mutations may be associated with attenuated disease, but certainly not all. So this is an example of a study on a pharmacoporon for Gaucher disease. They were investigating a pharmacoporon called N-nonal deoxynogeromycin. You may see that as a drug that is a name very similar to NDJ, which we also know as miglustat, which is a drug that's licensed for Zavesca for treating attenuated Gaucher disease. So these drugs that are evaluated as pharmacoporons, they're often what we call enzyme inhibitors. They bind the active site of the enzyme and they help it fold. at low concentrations, if they can get into the rough endoplasmic reticulum, they can actually stabilize the enzyme as it folds and allow it to produce a normal protein. So this is an example of 3 mutations, N370S, wild type, which is normal, and L444P. And this particular drug, N-nonal deoxynigeromycin, at low concentration, you see that peak go up. It's stabilizing that protein and letting enzyme be produced. At higher concentration, it just inhibits that enzyme, right? Not all mutations will respond. So this one, L444, it's always being inhibited. So this drug that's similar to NDJ, it got approved for another application. So it's not actually progressed to be used in this sort of approach. It's used to treat disease based on another principle. So is this an active area for the MPS disorders? And indeed it is. So there has been society funding for MPS 2, and there are also efforts afoot for MPS 4A and 4B. And there's a very good review from the lab that did a lot of the 4A and 4B work. But again, here's their caveat at the end from the abstract. Although the results show the potential for this strategy, further studies should focus on the development of disease-specific cellular models that allow a proper screening and evaluation of pharmacophorons. In other words, we need better experimental platforms. The ideal would be one that's extremely robust, rigorous, and what we call high throughput. It can screen thousands of drugs at a time. So one of the reasons we don't have faster progress in this area, part of it is just fate. Gaucher was a more common disease. There was a lot more work in the area at the time. Miglustat, which ended up being licensed for a different application but was evaluated in this particular approach, was already an existent drug. Many of these drugs that are being worked on for the MPS diseases, we need to produce them from nothing. They have to be synthesized by very, very capable chemists. It requires a lot of testing, synthesis, etc. It's arduous and slow. One of the things, however, is that we assume that these drugs are going to be active site binding motifs or enzyme inhibitors. That's not been rigorously tested. There may be other areas of the protein that we can target that might also have this effect, but how do you test that? And the big problem is we don't have a robust, rigorous, high throughput drug screen platform. And ideally that platform would be efficient, rapid, and it could test a diversity of candidate mutations. So the ideal would be you have a new mutation, in 6 months we have a drug screen for that mutation. And ideally it would involve cells that look like disease cells or even vertebrate models. And in the case of zebrafish, that kind of technology is potentially exists. We just need to get some work to create it. And again, this kind of robust approach would allow us to test thousands of drugs that maybe were approved as being safe, but failed when they were being developed for a particular application, what we call phase 3 trials. So one particular case I want to talk about for the membrane diseases, this kind of approach is theoretically a potential for patients who have membrane protein diseases, MPS 3C and ML2 and ML3. So if we've got a membrane-bound protein, when it's being produced, and this is a schematic from within a cell, we've got a ribosome, and on the upper left, it's putting that, shuttling that protein into the interior of the ribosome, and these are membrane proteins. So as they go through and get produced, they have what we call these fat-loving or hydrophobic stretches of protein that get parts of the protein stuck in that fatty layer. And it's those membrane-spanning fatty layer domains sometimes that get messed up during certain aspects of protein production for some mutations. So if there's a mutation in a patient that affects the development of these membrane-spanning domains for 3C or ML2 or ML3, these are potential targets for developing therapy. So it's one thing to say, in theory, you can do that. Is there any evidence from the animal model literature? So this is an example of an animal model in mice for something called hypogonadal mice. So these are mice that have teeny tiny little testicles and ovaries. And they have those because they have a GnRH receptor in the pituitary that has one of these mutations that means it won't get its membrane-spanning domains properly inserted, and so it doesn't produce a membrane protein. There are pharmacoporone therapies that restore fertility. So there are little drugs they've been able to administer to these mice in a pulsatile fashion, and you can see the effects. These are wild-type testes. These are testes from a carrier for this condition. These are testes for an animal that's homozygous for this condition. And this is an animal who's homozygous but has been infused with this pharmacoporone designed to restore normal membrane expression. So at least at a theoretical level, this is potential therapy for these membrane-spanning diseases, but you have to have a mutation that is affecting the maturation of those membrane-spanning domains. But still, for an area of our diseases that have not had a lot of potential therapies, I think there's a lot of hope in this approach. Existing hurdles, it still include de novo design and production of potential drug targets and getting a truly robust and rigorous screening process. I think in some ways, development of this may be, screening process may be one of our best options moving forward in this particular aspect of treatment therapy. So that's chaperonins, and they can also be used as drugs that will chaperone enzyme therapy. This has been talked about for various ERT applications. There have been some clinical trials or at least drug pipelines, but nothing's come from it beyond that. Stop codon read-through. This is the last of the approaches that will actually influence some aspect of RNA production. So if you have a stop codon, there's a system in the body that gets rid of most of those transcripts. It's called nonsense-mediated decay. So a little stop codon there in that second panel, so to speak, will lead to destruction of your message. If you can develop a drug that will allow the ribosome to read through that stop codon, substitute it for another amino acid, you could get some small amount of protein. And there are antibiotics, we know, a class called aminoglycosides, that actually bind ribosomes and have this effect. And it's a hot area of research. Nothing's come to clinic yet. Two, as it were, more common rare diseases, cystic fibrosis and Duchenne muscular dystrophy, really dominate the research area of this particular approach. And there's been no success yet. When there is, though, it will have almost immediate hopeful application to the MPS community, in no large part because iduronidase alleles, that's a misprint, it should be W402X, and the other allele Q70X. Those account for a large percentage of our MPS 1 patients. So I'm not going to say more about stop codon read-through. I don't know that there's a real place for the MPS applied research area. There's so much more going on with companies focused on cystic fibrosis and Duchenne muscular dystrophy. I kind of figure we should let them do the heavy lifting in that regard. So let's shift focus as we wind up and onto pathophysiology disease processes. So substrate deprivation has been validated in mouse models of MPS 3A by a group from Jeff Esco's lab, where they actually crossed MPS 3A mice with mice that were genetically incapable of producing heparan sulfate. And indeed, that approach works. There's a limited literature. The only place I know about work being done is Dr. Ainsley Derek Roberts in Australia, and they used rhodamine B, which you may know of as phenolphthalein, and they've been able to treat both MPS 1 and 3A with it and get benefit. It appears safe in mice, even across generations. It reduces GAG and ameliorates phenotype. This has not been pursued further. Perhaps there's a priority to try and evaluate this in a larger animal model. Moving on to from substrate deprivation to treating pathological cascades of disease. This is one of my favorite topics and one that I think is most exciting. So this will be a chapter where we focus on work done in MPS 6 originally with a drug called pentacin polysulfate. This work was spearheaded by Dr. Leila Simonaro, who's on our scientific advisory board. She validated the approach of using a drug called pentacin polysulfate in MPS 6 rats. This work has been repeated in murine MPS 3A as well as canine MPS 1. And it really capitalizes on the anti-inflammatory effects of this drug, or at least that was the entree that made Dr. Simonaro Leila think about this. So PPS actually is a drug that's used as an anti-inflammatory, and Leila and her colleague Ed Chookman knew that these diseases had inflammatory markers. So they were looking at this drug to try and interrupt that process. But as you can see in the rat study, this is a normal skull in the top right, or excuse me, top left radiograph. Middle top radiograph is an untreated affected skull from a MPS 6 rat. And on the upper right, that is an animal just treated with a drug. And look at the changes in the bone structure of that animal. When I first saw this, I was gobsmacked. This was a really impressive result. And I was really excited to see Leila pursuing this work. And it wasn't just anti-inflammatory, because we saw decreased GAGs in her study using the MPS 1 dog. So the panel in the upper right, these are decreased levels of GAG in animals treated in the white columns orally and in the gray columns with injected PPS. The gray or the hash line down there is normal, but this is a drug that's only supposed to be anti-inflammatory. How is it reducing GAG in some cases by half in the kidney? So this was really exciting. Leila went on to show that markers in the brain, this is the panel on the left of MPS 3A mice that were treated with this approach, markers for lysosomal storage in the brain were reduced in treated animals. So this was just an anti-inflammatory drug as far as we knew. Somehow it's reducing substrates in the periphery and markers of storage in the brain. So I think this is a really exciting approach to therapy. Proven anti-inflammatory effects, unexplained positive effects on GAG storage and lysosomal markers in the brain. And with this decrease in storage, there's a potential strong therapeutic application. And indeed, this drug has already gone through a limited human safety trial in MPS TRUE, and I don't have anything more to report on that, but I'm sure that we'll see more to this story in the future. So other approaches to treating pathological cascades that may be on their face sort of symptomatic, but I think may also yield impacts as we watch them, if they get approved, be adopted for a more routine approach. So there are two studies being done by Dr. Linda Polgren. One involves Humira, the other involves anacrina in MPS 3. The first is MPS 2. They're looking at progression of bone pathology and behavioral changes in MPS 3. So will that affect long-term progression? Hopefully. Those are active clinical trials that Linda's involved in. We also know that there's a drug therapy of bisphosphonate for mucolipidosis. Really, it's treating pain. It's sort of symptomatic. I wonder, as this is used for longer term, whether there's impact on the progression of disease. So altering vesicular trafficking, I'm going to try and speed up because we are getting close to time. This is an approach where we're actually trying to get enzymes to get turned over faster, lysosomes turned over faster. It's an area that was discovered by Andrea Ballabio, and there is a very important approach involving trehalose in mice that's been shown to be effective in MPS3, and there's now a clinical trial being planned for that in San Filippo. There was rare orphan disease drug status was just approved for that. And then we have the what the heck drugs. So that the example of that would be cyclodextrin treating MPS or Niemann-Pick C. And that story, I don't have enough time to go through now, but it was really kind of stumbled on that this drug carrier actually influences this disease process. Do we have something similar going on in MPS disorders with PPS? Maybe we do, not sure. I don't have time for questions, I'm afraid, and I need to get over to another session, so I am going to complete this. We'll get to your questions sometime in the near future. Thank you.